is my lab life in Oman. This is how we receive the sample. Sample is barcoded or received through patient ID. Then the rest of the request for that test for that particular patient will comes out. So this is the one. Okay, this is our reception. This is the receiving area in the reception. This is a safety cabinet 2 where the sample are processed inside the safety cabinet. Inside is the negative pressure and outside is the positive pressure. So all air is coming from the outside going inside. So there's no air or coming out from this safety cabinet. That's why it's become negative pressure there inside. This is the pneumatic tube. Okay, all samples received in the wards are given through this pneumatic tube. There's like a bucket. They're putting it in a canister or bucket. Then they are loading it in this. I think this 8 inches. I think some samples are coming. I heard some samples coming. No, maybe no. Okay. So this is the reception. This alone is microbiology. This is the urine section. This is the Copus 411 where we do the test for urinalysis and urine microscopy. This is the centrifuge. This is the fume hood for stool. Okay. Refrigerator. This is wound section. This is also a safety cabinet 2. This is where we process also all infectious materials inside. Same principle with the other one in the reception. So if you hear the air sound, it's going up, circulating, going up. See? Safety cabinet. You can see. So this is the SO Infinity. Sash in the same position. So meaning it's working properly. This is the controls. Okay. This is our microscope, Olympus microscope. Okay. This is the carbon dioxide incubator. It has a maintaining of 7% carbon dioxide and the temperature inside is 37 degrees. Okay. This is the reading plate. Today morning, 24 hours. We're stocking it and we're throwing it after 72 hours. Okay. This is sputum section. Same. There's a microscope there. This is the new machine for COVID-19. Still under evaluation. This is the gram staining machine. This is another safety cabinet where we are processing the COVID-19 samples, swabs. Those are negative swabs. Okay, we're putting it in one biohazard bag, then later on to be autoclave. This is the pending test that I'm processing now. Okay. So this is the safety cabinet unlatched. So there's a like still need to adjust it later on. It's like loose. Okay. So this is also a safety cabinet 2. I'm going to fix this later on. Maybe it's only loose, the latch. I don't know what happened. This is the Gene Expert machine. This is where we are loading our COVID samples, COVID test kits. Yeah, like this one. It's already gone out. It's already finished. So we need to release this one. Okay, this is the Gene Expert cartridge for COVID-19. We're keeping it all together there, and we are autoclaving it afterwards. So this consists of 8 tests. We can load like 16 tests all in all, but I think the upper top is not yet allowed to open, so we're only loading 8 samples at a time. We cannot load the 16 samples. We still have to open this one. So after coming out, the result will be reflected in this computer or transmitted in this computer. You have to enter the pass. So that's it. So we'll view the results. What is the results of the patient report? There are so many reports here. So it's negative. The result is negative for that patient. Yeah, it's negative. So we have to print this and release the report. We have to release the report. It's negative anyway. Okay. This is the blood culture section. This is a category 3. See, there's a, you can't open it without this is a negative room for category 3. Category 3 hazard, group 3 bacteria. For bacteria, there are like categories, how dangerous or potent is the bacteria. So we need to like process in a more isolated room. So this is the safety cabinet 3, where we are processing blood culture, infectious materials. Although safety cabinet 2 is also good, but this is more safer. Of course, this is a class 3 cabinet, so this is more safe. This is a biosafety class 1 cabinet, but they convert it to safety cabinet 3. So there's an exhaust there, negative pressure also. This is the Bactec FX. So as we see, there's no positive today. If it's positive, that green color will become red. This is the carbon dioxide incubator, same temperature, 37.7% carbon dioxide. This is the aerobic incubator. Okay, it's also 37 degrees. This is the Campi incubator. If you see that the temperature is 43 degrees. So we have to maintain the 43 degrees, 43 degrees for the Campylobacter to grow. Campylobacter organisms like Genie, coli. This is the old Bactec 90050. This is microphage. This is all the same plates. We have also here gram stain machine. We're using this one for gram staining, but right now we're doing the manual. I have to go out now. I have to show you each room, which will be. I'm going inside of Autoclave Room. This is where we are throwing our COVID samples for autoclaving. Right now I'm only collecting garbage. I am going to autoclave the wastes. So this is the 4 autoclaves that we are using in autoclaving. 2, 3, 4 for our wastes. Lots of mess on top. It's not supposedly, it should not be. So this is the media room where we are doing preparation of the media. This is the master cave where we are doing preparation of the media. This is the dispenser, and that is the printer. This is the water bath incubator. I think they're using it for Rosella now. So this is the media room. This is the serology room. Lots of files. This is the architect machines. They're loading here all serological tests. Not all, but almost all serological tests. Hepatitis, measles, rubella, HIV. There are some tests here. They're loading different tests there. I'm not very familiar with serology test, but this is also another solution. This is the new machine. I think so. This is there for validation, where they're doing ELISA. And now, yeah, I have to show you the electron microscope. Yeah, this is the electron microscope. Yeah, it's the electron microscope. This is another electron microscope. Oh no, fluorescent microscope, sorry. This is the fluorescent microscope. It uses a specialized stain in order to view the fluorescent antibody or antigen. There's a special reagent for this one. From the word fluoresce. If you view it under this microscope, it will fluoresce. Like, it depends on what stain you're using. Some will stain yellow, red, green. It depends. Depends on the type of fluorescent stain you're using. So this is the fluorescent microscope. It has a filter. They're putting here filter at the bottom. Specialized lens. There's a adjustment here also. Depends on the wavelength of the UV. Okay, so that is the fluorescent microscope. This is our stock room where we put our stocks. Same thing, this is our centrifuge, another centrifuge. We'll go to, this is another machine for ELISA, immune analyzer. This is for ELISA also. We're using this for other tests also. ANCA. So that is the another machine. And this is our ABS section. We are doing here identification and sensitivity of microorganisms. This is the BD Phoenix M50. This is new, just newly installed. This is the old one, but same BD Phoenix M50. We are loading here the organism. After suspension, we are loading the organism here for identification and sensitivity. We are using a special panel for that one. This is like the automated turbidimetry for measuring 0.5 McFarland. So it's an automated. We're only loading here the sample from this. We're only loading the sample from this tray, suspended colonies. Then we are loading this machine. This is BD Phoenix AP. It's like nephelometer, automated nephelometer. And this is the MALDI-TOF. This is also used for identification only, not for sensitivity. It can identify the organism within like 15 minutes, 10 to 15 minutes, using mass spectrometry. The principle of this one is different from this BD Phoenix. This is more of colorimetric, but this is more of mass spectrometry. This is a combination of, I think, colorimetric and spectrophotometry also. But this is more high-end. It can detect for as fast as 10 to 15 minutes. This is the MALDI-TOF Biotyper MBT Smart. So this is the ABS section, and I will show you the cold room. Yeah, this is the cold room where we are stocking our stocks that should be maintained 2 to 8 degrees centigrade. It's very cold here. Not very cold, it's 2 to 8 like a refrigerator temperature, only you are inside like a big refrigerator. So we're putting our plates. We're using these plates in culturing different media or different organisms. So it's quite mess also. Sorry for that. So this is the Serology Reagents. This is the ready-made media. It's already packed from our supplier. This is different. This is each box is different. Not all are the same. So this is the media room, the cold room, rather. Okay, I'm going to close this. This is maintained 2 to 8 degrees. This is now 2 degrees. As you can see, this is now 2 degrees. Temperature inside is 2 degrees. I didn't show you the hot room. This hot room, the temperature now is 36.6. 36.6. This is the hot room. Yeah, where we are incubating our subcultured samples or bacteria. So each this is for urine. We are incubating the bacteria. I don't know if there's any growth now. Yeah, see, there's a growth of colony there. One colony is equivalent to 1,000 bacteria colony forming units. We call it as colony forming units. So this is where we're incubating the plates. This is maintained 37 degrees, 36 to 37 degrees. Yeah. Now, because I open it, it's only 35.2. So that's it. So I have here an anaerobic jar. I'm going to close this. I'm waiting before 12, and I'm going to close this one. I'll put the sample inside. This is for anaerobic culture. We're putting a gas pack for this one to maintain the anaerobic environment inside. Then we're putting it in 37 degrees. Okay, that's all, folks. What do you want to see in this tab? This is where I'm working in microwave for almost 7 years now. So later on, I'm waiting for some COVID samples. Then I will load it here. So this is the kit we're using. SARS-CoV-2. Okay. This is the kit. Inside is like 4 compartments there for extraction, for elution, extracting the RNA, then eluting it, then transcription. There's a transcription going, then measuring the CT and E values. Okay, so that's it. Later on I will autoclave this one. It's already almost full now. Okay, that's it, guys. Bye.